So this part, this whole thing, a pathogen, there's this little antigen. A white blood cell would come in here and eat it and pull it inside. And then, once it had it inside, it would take the antigens off. It would break it apart. And this macrophage could then take those antigens and display them on the surface, like that. So first it takes it in, then it tears it apart, engulfs it, destroys it, and then presents it. This is extremely important to immunity, okay? Because we know that there's the 3 R's. What are they? Recognize, respond, and remember what? By what? By the antigen. So really, it's really remembering, responding, and recognizing the antigen. You know what? Most of the time, our white blood cells, like lymphocytes, you know how they recognize it? Because it's being displayed by what kind of cell? An antigen-presenting cell. So they will come over and contact this, another white blood cell here, make it a lymphocyte. How do we know it's a lymphocyte? What am I trying to do? Come on, use your big brain. You remember. A big nucleus. Yeah, right? They have a big nucleus. Big, huge, round nucleus. Just a sliver of cytoplasm outside the nucleus. You remember that? So it comes over, it contacts it, it links up with it, and it goes, oh, here, right here, is the bad guy. This little arm that comes out here would say, oh, I recognize this is a good guy. Good guy and bad guy, by the way, are not technical AMP terms. Just understand that. I'm speaking to give you some point of reference right now. So antigen-presenting cells present the antigens to what? Right, and specifically which other cells? No, the macrophage itself is known as the APC. Very good. The lymphocytes, right? It's presenting it to the lymphocytes so that they can go through the processes that we consider specific immunity. See, this sets the stage for it. They have to have the ability to recognize, respond, and remember. And that's all about the antigens. It's all about them being displayed. Okay. So far, we have said that the antigen-presenting cell is what kind of cell? A macrophage, which is a white blood cell. But I need you to know that there's another kind of cell that more commonly functions as an antigen presenter, and it's called a dendritic cell. Dendritic cell. Dendritic cells are another group of white blood cells. They're called dendritic cells because they have long arms. They are the most common antigen presenters. I've got comfortable talking about macrophages as antigen presenters because I think by the time people get to AMP2, if I say macrophage, you're real comfortable with the word. And I like to talk about things you're already comfortable with because that involves less memory. But the truth of the matter is most of the antigen presenters are dendritic cells, even though macrophages do this too. So it's more correct to say that. You know, you've already heard of a dendritic cell. In AMP1, you've heard of Langerhans cells. And those are a type of dendritic cell that lives in the skin. But when we're in AMP2 and we think Langerhans, we think of something else. So kind of stay away from that term. Okay, back to our opening slide. The 3 R's are the ability to what? Recognize, respond, and remember. And what are they recognizing, responding, and remembering? Okay, a pathogen through its what? Antigens that are usually on the surface. How do those antigens get displayed? By what kind of cells? The perfect answer is antigen-presenting cells. Name two types of cells that function as antigen-presenting cells. Dendritic cells and macrophages. So hopefully already you can see that this is not easy as heart anatomy. This is small. This is invisible. We can draw cells, but they're all going to look the same because they're white blood cells. They're going to look very similar to each other. So this is more informational. This is more small. This is more tiny. It's not something substantial that you can put your hands on and really understand in the same way that you could by looking at and pointing to a lump. All right? See, the immune system, when we get to specific immunity, we're talking about cells more than we're talking about organs. We're talking about white blood cells. And we'll be talking about the chemicals they secrete as well. Okay, now, when we talk about immunity, there are two basic categories. There is active immunity and passive immunity. Active and passive immunity. Look at the little picture up here at the top. From what we've drawn so far, can anybody tell me this little thing right here with its little stick there and this one right here with its little stick there, what are those supposed to be? Pardon? Okay, those are the pathogens. And what's the little stick part that's going in here, the little splinter looking thing? What's that? That's an antigen, correct? That's an antigen. Okay, I'm going to talk about B cells for a second, even before I go to this active and passive. Bless you. Okay, so back to this. Here's our little pathogen. Here's its little antigen. Here's its little antigen. What I'm showing you here is after the immune response has happened, our B cells make these little chemicals called antibodies. Yeah, we've all heard of antibodies, but when we've heard of antibodies, we've heard about the antibodies in the blood that are there for blood types. Even though they're similar chemicals, that's really quite different than this. Your B cells or B lymphocytes, the way that they fight the pathogen is B cells fight the pathogen by releasing antibodies. And a generic antibody always looks like this. It looks like a Y. And then it's got two sticks on its arm right here. We're going to talk about this tomorrow as well. So you see this is the antibody here, a little Y down there. Here's an antibody here, and here's another one here. So you can see that they kind of resemble a Y. Antibodies are made to attach to whatever antigen they're supposed to go out and get. Okay? They attach at this one little region, which we'll talk about later. Don't need to name it or anything for right now. But then you'll see the rest of the pathogen would be attached to it when it goes out and links up with it. So when we're talking about active and passive immunity, what you're going to actually see is we refer to this in whether the immune response is actually making antibodies. If we are making antibodies, we call that active immunity. If you are making the antibodies, we call it active immunity. What cells make the antibodies? Very good, B cells. Yes, tomorrow as we get more in depth, I will change that. But it is a subset of B cells. B cells actually, when they get activated and divide, they call them plasma cells, and plasma cells make the antibodies. The point is, they still come from your B cell type of immunity. Any pathogen that's harmful to our body that our B cells would in turn make antibodies against, we could say that we have active immunity taking place. If our bodies are stimulated at all to make the antibodies, that is active immunity. And there's many situations that cause that, and we'll talk about them in a second. We also have a type of immunity that's called passive immunity. Okay, if active is when you're making antibodies, what would passive be? You're not making them. You are receiving them from another source. This is when you receive antibodies from another source. That's a little different. Because of the name, it would sound like that's it, but it's really not. Okay, now here's the fun part, guys. Let's separate active and passive right down the middle. There are two types of each. There's a natural version of each, and then there's an artificial. Let's do the natural first. The natural way of things means not medical. Okay, every time you see natural, it means not medical. It means through a normal life, not involving medicine, what would occur. So for active immunity to be natural, that is the most basic, the most simple thing that there is on the planet. You come to class, your neighbor sneezes on you, the pathogen gets inside of you, your body makes antibodies to fight that. The active part is you making the antibodies. The natural part is due to a normal exposure. And you can see that right there. Making antibodies due to a normal exposure is called natural active immunity, or you could say active natural. I usually say natural active. Well, that makes sense. You go to Taco Villa in the 1980s. You eat a burrito that has lettuce on it. That lettuce was cut on a cutting board that had chicken on it. The other one, the Shigella, got in it. And everyone in Odessa that ate there that day got shigellosis. Massive vomiting and diarrhea and other pleasantries that we won't talk about. That was natural active immunity at its best. Normal exposure, going to eat somewhere. Harmful stuff, pathogens were in the food that you ate. Your body responds to that. Part of the response isn't what we would necessarily like, but it's trying to help you. On a cellular level, what was going on? Your B cells in your body would have been making antibodies and dumping them into the body fluids to try to fight that. And that would be a natural active type of immunity. Artificial active is very common nowadays. Most people have this taking place in their bodies. Remember, artificial is always due to medical conditions, interference, activation, however you want to look at that. The artificial way to activate your immune system and cause your B cells to make antibodies would be to inject that foreign invader into your body to stimulate your immune system to fight it, to cause your B cells to make antibodies. We call that a vaccination. See, when they vaccinate, they try to make the pathogen harmless. They say they attenuate it. They either kill it or they make it to where it can't cause you to get the disease. But pretty much most people now know that sometimes that doesn't work, and people get a vaccination for the flu. Quite frequently get the flu. Okay? Sometimes they get the same strain. Sometimes they get different ones. They work, but sometimes they don't work as well as they're supposed to. Okay? That is active artificial. Natural, normal exposure, you make it. Artificial, medical exposure, you make it. What's the it that you make? Antibodies. And these antibodies go out through your bodily fluids, and they would attach to the antigens and neutralize them. And we'll talk about more how that works tomorrow night. Passive immunity. The antibodies are received from an outside source, another source. The natural way to get them would be to get them from your mother. There are two separate conditions that cause you to get your antibodies from your mom. Number one is just when you're growing inside of her. Her antibodies, you're in that nice, warm, comfortable fluid environment floating along, and antibodies can cross the placental barrier. The smallest antibodies can do that. We'll name them tomorrow night. That's a natural, passive way to get your immunity. Doesn't last very long, lasts for a few months. Because once you're outside of mom, her antibodies aren't coming through you that way anymore. Then you're outside of mom and you get hungry and you got to eat. And the natural way to feed a baby is for the baby to breastfeed. So, pretty cool that there are antibodies in mother's milk and breast milk. Baby drinks breast milk, baby gets antibodies from mom that way as long as the baby is breastfeeding. So if you got a kid that breastfeeds till he's 2, he's getting an immune system till he's 2. As soon as you take him off the breast and put him on the bottle, no longer getting antibodies from mom. Okay? But you're not getting bit either, right? Yep. So notice, both natural passive types you receive from mom. Sometimes when you're inside of mom, sometimes after birth. This is one of the advantages of breastfeeding to the baby, is it helps to keep the baby's immune system stronger. Okay? Because at this point in time, the baby's immune system is developing early stages. So they've shown, sometimes they show that babies that get breastfed can be healthier than babies that are not. Okay, what would be artificial? Artificial would be when the antibodies are injected into a person. That's different than a vaccination. In a vaccination, you inject the bad guy or the antigen, the part of the bad guy, and the body still produces a response. With an antibody injection, what happened is they probably injected a horse, and the horse went through the immune process and produced the antibodies, and then they pulled the antibodies from the serum, from the plasma of the horse. They took them, and now they'll inject them into people. They do that. They use horses. They can use other animals for that. And now they can even grow antibodies in labs. They can fuse tumor cells with B cells and do all kinds of cool stuff and grow those antibodies in labs outside of living animals and do that. This would be like a gamma globulin shot. A gamma globulin shot would be an example of artificial passive immunity. So it's going to take a little time to get those straight. I always ask about differences between natural, artificial versions of both active and passive. You're going to have to identify what goes where. So just make sure you learn all 4 of those. Let's talk about B cells first. The type of immunity produced by B cells is more appropriately called humoral immunity. It starts off with one of these. Yes. A nice little, sure, it could be a macrophage, it could be a dendritic cell. Its big name is antigen-presenting cell. It takes in the bad guy. It tears him apart. And it displays, somewhere on its surface, the little antigenic determinant. On what kind of MHC? Are you sure? It's an MHC2 because it's an antigen-presenting cell. Okay? Right? Antigen-presenting cells, mostly MHC2s. It's all body cells that have the MHC1, but antigen presenters specifically have MHC2s. Right? Because we're not trying to kill this antigen presenter, are we? We're trying to go out and kill the bad guys wherever they are. So look, a helper T cell comes over. Oh, the helper T might be bigger. I just draw him like he looks like a little cute baby. It's a helper T cell. Now, we'll add a little piece of the picture. This little way that the helper T links up it gives the helper T another name because they call that little linker protein a CD marker. They call it a CD4. CD stands for cluster of differentiation. I don't think you want to memorize that. Okay, so we'll just call it CD4. So sometimes they call helper T cells CD4 cells. Just another name for them. Our book talks about it a lot. That's why I'm mentioning it to you. Do helper Ts kill things? No. What do? They work to stimulate the rest of the immune response, specific immunity, by releasing chemicals called blank. Very good. Cytokines. Here they are. Many different cytokines. We're not getting all into that. If you ever take immunology, you'll have a blast with it. Now, these cytokines can activate a B cell. And when they activate a B cell, that B cell will only be capable of fighting against what? That one particular or specific AD. Right? That's it. Can that one specific particular AD be on 20 billion cells? Yes. It can be 20 billion bacteria in your body. As long as they're that same one with that same AD, this particular B cell can fight it. What if it's a different bacterium or a different surface marker? Will this B cell be able to fight in any way? No, it would have to have a whole other separate population of B cells prepared to do that. Okay, so this B cell, those cytokines get over there. And remember, most of the time we say that this B cell has already had a prior contact with what? The antigen. Could we say the AD instead of antigen? Sure, we could. We could say that. So this B cell's ready. The cytokine hits it. The B cell is now active. Here's new information. We haven't talked about this. The B cell will clone itself, a huge population of clones. So they rapidly divide. Notice how I'm kind of separating them out here. Most of these B cells will get a new name. And they will get the name plasma cells. And those plasma cells will do this. What is that? That is an antibody. These plasma cells will crank out antibodies at the rate of 2,000 per second. An active plasma cell, can produce 2,000 antibodies a second. That's 4,000 in 2 seconds. That's 120,000 in a minute. Every minute. How many is that in an hour? A lot. Wow. 7.2 million. It's crazy numbers. It's a lot. Every hour, it keeps crunching them out. What do these antibodies go do? They go find an AD and bind to it. And we haven't talked about how they work yet. I'm going to get to that rather quickly. Look, when we cloned these and went through clonal selection, remember that word from last night? Clonal selection. SELECTION. This is supposed to be an A right here. Some of them become plasma cells, and what are these over here? These are memory cells. Memory cells are not capable of making antibodies yet. What's a word that means they can't make them yet? Pardon? Not a fancy word we learned tonight, just a word that means they have to sit there and not do anything for a while. They lie dormant. They're dormant. They're inactive for a while. Usually it's a few months. There's not an exact number of hours, but they are inactive for a while. And the only thing that those memory cells can ever respond to would be what? That one specific antigen. Could you ever meet up with that one specific antigen later in your life? Yes. If that bacterial strain comes through again and people get sick with that same thing later in life, you bet it can come through again. And this person later in life would be protected from that. See, when memory cells get stimulated later on, they produce a larger response and a quicker response. Much larger and much quicker. In fact, the first time you meet a new antigenic determinant and you go through the B cell response and you make antibodies, it takes 7 to 10 days to really combat that. They call this a primary immune response. That's the very first time that you've ever met that bad guy. And in a primary immune response, you will actually get sick. Because the bacterial population or whatever will grow fast enough that you get sick, then your plasma cells start kicking in after some time, and about 7 days in they reach maximum and it starts to fight it. But with a secondary immune response, we'll just put IR for immune response, that's quicker. In just a few days. It could even be 1 to 2 days. They reach maximum intensity of production of antibodies that quickly. And so commonly, with a secondary immune response, a person might get just a little bit of sniffle or just a little teeny bit of a sore throat, and they never really get sick. You're like, oh, I thought I was getting sick, but I didn't get sick, and I'm better. That's because the secondary immune response is faster and it kicks in, right? And it used memory cells. Well, if it's a secondary immune response, are you going to use all the memory cells, or are you going to save some of them for a third time? You're going to save some, but guess what? The third time you'd still call it secondary. The fourth time you'd still call it secondary. The fifth time, the 100th time, it's still secondary. So remember, when memory cells get activated, they won't all get activated. Some of them will become what cells? What are the active ones called? Plasma cells. And some of them will stay memory cells. So while you're thinking about that, look, here's a slide that shows it. Primary immune response. The first encounter with a new antigen. B cells get activated. They proliferate to form a clone. Some of those clones become plasma cells, and some of them become memory cells. Cool. Secreting some antibodies. Lag period. Peak levels over a week, about 10 days, then they decline. Secondary, faster. Peak levels a few days. Look, here's the picture that shows it. Here's a primary immune response. Takes a few days to really start producing them, and then they kind of kick in. And they kind of fall out. Look at this secondary immune response. Wham! Look how much stronger it is. That's like 3 powers is like almost 1,000 times stronger. I won't ask you 1,000 times stronger. Just a lot stronger. And look, the levels stay up because it really wants to fight it. Here was the primary immune response. It only maximum peak right there, and then it's dropped off. This one, peak way up there, and then stays up high for longer. This humoral immunity that we're talking about with the B cells fighting by using antibodies, that's fighting against a foreign invader that's in a body fluid somewhere, right? Spreading through the interstitial fluids maybe. And no, the B cell that becomes active that makes the antibody is called a plasma cell. Okay. Yes, remember, helper Ts, they don't just help T cell responses, they help B cell responses too. Humoral immunity has two parts, active and passive. Active can have both a natural and an artificial, and passive can have both a natural and an artificial. So there are a couple of slides right at the end of PowerPoint 1 that if you want to take some time and see that in a more organized format, then there it is. Okay. 21b. And it starts off by talking about antibodies, which makes sense because we were just doing what kind of immunity? Humoral, that involves which cells? B cells, and they become activated and called plasma cells, and they make what chemicals? Antibodies. Antibodies are proteins. Antibodies are proteins. When they're in the blood, they're called immunoglobulins. Well, they're always called immunoglobulins. But in the blood, they're part of the gamma globulin portion. Remember how we had plasma protein? Some of those are actually antibodies. Immunoglobulins. Why would we call antibodies immunoglobulins? Globulin tells us it's a protein, and immuno tells us what system? It's part of the immune system. Don't you like how some stuff in AMP makes sense? And don't you like how we use stuff over and over? If I would have said globulin one time and that's it, it would be a little harder than if we keep talking about them. And we've talked about them multiple times. These proteins are secreted by what active B cells? Plasma cells. And what are these antibodies searching for when they are sprayed through the body fluids? They are searching for ADs, otherwise known as antigens on a specific bad guy that we might call a pathogen. Right? Oh, you should have. You should have said that. Basic antibody structure. Oh, we just have to draw them a little bit. But let's look at this picture first before we get in there. Look here. It looks like a Y. You've seen me draw the Y. Look, I draw the Y here, and then I put these two little arms sticking out right there. If this is a protein, that means that these are big amino acid chains. Okay? These are the big ones down the middle. These are the little ones on the side out here. Right here, this portion here, is the antigen binding site. That is where the little antigen will bind to. By what? By shape. By what we might call lock and key. So this shape right here, they're not showing us very well, but the shape of whatever this is very particular and important. This down here, this bottom part, it's known as the constant region because it doesn't vary for a specific class of antibody, and you'll see that in a second. Up here where the antigen binding site is, it's the variable region. And that doesn't mean in one antibody that it would vary. It means that the different antibodies, the way that they're different is by this part right here. That's how they can, why this antibody can bind to antigen A and the other one could bind to antigen B because of this variable region right here. Now let's get real fancy and fun and technical. If an antibody is a protein, doesn't it have a specific shape? Do you remember from AMP1 that proteins have specific three-dimensional shapes? So when it's built, this area right here, this variable region or this antigen binding site will have a very specific shape to it. And its shape will be made to fit what? The bad guy. What part of the bad guy? the AD, the antigenic determinant, right, Sherry? It would be made to fit that. That's how it works. So what it does is it neutralizes them. It grabs onto them and it renders them ineffective, or does some other things which we'll see in just a second. So when we draw antibodies, oh boy, we draw them. Now you see why we draw them like this. Look, I draw one up here in the corner for you. Here's the constant region. Up here would be the antigen binding site. We don't really abbreviate it that. Anti-lock braking system. Antigen binding site. We call that portion or that little region right there variable, because it can change. So look, if I had a bad guy, we'd like to draw him like this, then it would get grabbed a hold of right there, and this one would get grabbed a hold of right there, and it would render them ineffective. Really, antibodies are pretty simple. They're chemicals that go out and attach to stuff and kind of neutralize them or cause them to fall off into a solution or do these other things that we're about to describe. Remember, these are dumped out in massive quantities. 2,000 a second by a healthy plasma cell. Wow. So they can go attach to lots and lots and lots of antigens. Now the fun part. There are 5 major classes. 5 major classes of antibodies. Here are the 5 classes of antibodies, also known as, we know, immunoglobulins. Right? Antibodies are immunoglobulins. Their name always begins with a big I and a little g for immunoglobulin. And their name is just, after that, it's just one letter. They're known as IgGs, IgAs, IgMs, IgEs, and IgDs. There are just 5 different classifications, 5 different classes of antibodies. So what we want to know is we want to know just a little bit about each one. Now look at your PowerPoint. I left you this stuff right out of the book that describes each of them in depth. Okay? That's cool. And then the PowerPoint kind of summarizes it. But I like to let you see it handwritten like this, because I summarize it real short and sweet and to the point. You know, when you're taking notes, fewer words is sometimes better, isn't it? Because that's how our brain, it can lock it in. The most common circulating antibody, somewhere around 70% of all of your antibodies are the IgGs. IgGs happen to also be the smallest. They're the most numerous and they're the smallest. They are so small, in fact, that they are the only ones that can normally cross the placenta. So when we talked about naturally acquired passive immunity last night, we were talking about IgG antibodies coming across the placenta from mom into baby that helps to give the baby a good portion of its immune system for the first few months. Antibodies are just chemicals, though. They're not cells. They're not living. They're just chemicals, and they get broken down over time. So these antibodies only protect the baby for a few months. But then the baby gets out, and the baby has to eat, correct? And it drinks mother's milk, hopefully. And in mother's milk, there are IgA antibodies. Look here. See this? There's the bottom of one, right, of this one, and the bottom of this one. See how they're joined right there together like that? IgA antibodies are twice as big. They're like two antibodies stuck together. So they can use the term, they're a dimer. That means two units, two subunits, right? You've heard of monomers and polymers from AMP1, repeating subunits. A dimer is two repeating subunits. It's an antibody that's twice as large. So check this out. How many binding sites for bad guys, for antigens? 4. You can see that. It could actually bind to 4 things instead. That's cool. A little bitty baby that's drinking mother's milk that doesn't have a lot of immune system, the mom feeds the baby IgA antibodies in the milk. By the way, IgA antibodies are also in your tears and your little secretions from your nose and saliva and other bodily secretions as well. But look, they have twice as many binding sites. Isn't it cool that the immunity that mom gives the baby after birth provides the baby with twice as many binding sites to help make up for the fact that it doesn't have a huge immune system yet? I think that's pretty neat. What's this? Yeah, good. Pre-milk, right? The first day, mother makes that milk. It's a little milk, but it's mostly an immune system cocktail chocked full of antibodies. So even if you're not going to breastfeed your baby through a long period of time, it's really good to breastfeed your baby for a little bit, especially the first couple of days to get that. Okay, now this is pretty neat. The IgMs stand for mega. IgMs are actually 5 antibodies. I'm not going to go crazy and draw that one. I'm going to show you. Look here. 1, 2, 3, 4, 5. Right there. So how many binding sites? 10 binding sites. You don't have to draw it either. But you can. It's fun. Twice as many binding sites would make sense in a primary immune response. A primary immune response is the first time you meet a harmful invader, right? A pathogen, an antigen. Your plasma cells are smart. In a primary immune response, we know the bad guys got the jump on us because it takes so long to build them up. So what does it do? It produces antibodies that have 5 times as many binding sites as the normal ones. It produces bigger ones. So can you imagine how much longer our primary immune response would take if they were all little bitty antibodies like this? 5 times as long. I love the logic there. IgE antibodies, now these are crazy. These are the ones that cause your symptoms of allergies. They're not helping you, they're giving you allergies. These are the antibodies that stick in the back of your throat and in your nose, and they activate white blood cells that cause histamine to be released. Oh my goodness. You know, they're there for a reason. We call it an allergy when they're overproduced. When they're being produced against something that is normally not harmful, like a hapten, some small molecule that then binds with us and then causes this to occur. And the last kind, IgDs. IgDs are basically B-cell receptors. You notice when I'm drawing these macrophages and B cells and stuff, I'm drawing their receptors looking like this. B cells are smart. If they have the capacity to make antibodies that they dump out there, they can just, since it's a protein, they can just stick it on its surface and it can function as a little receptor for something as well. So that's cool. They don't know much about the IgDs, or at least they didn't when I started teaching 400 years ago. I'm sure they've learned a little since then. Okay. If you want to remember a list of 5 things, what's the best way to do it? Make it spell a word or a sentence. But to me, this is great work. Look at the order. Gamed. You ever been gamed? So there's other things that you could spell too. Like if you want to go to A&M and you want to be a doctor, you could say AgMed or DIMOG or, I don't know, edamame with a G. I'm just kidding. But anyway, the whole point of the matter is, you come up with one of yours. So how do antibodies work? Antibodies work by having a plan. And we're going to use pictures and handwriting. Plan. That's an acronym. And the other one that you're going to have, that you should have from listening to non-specific immunity and taking great notes, is how complement works. And complement's memory device was, anybody? Yeah, where's your iPad? Oh, Pickle. Do you remember? Oh, Pickle? Complement works by causing phagocytosis, inflammation, and cell lysis. Anything that is a marker that tags something for phagocytosis is called an opsonin. An opsonin is anything that marks something to be phagocytized. So when an antibody comes and binds to an antigen, one of the things that happens is that causes other white blood cells to gobble it up. Antibodies that are hanging around, bound to stuff, our white blood cells know to eat those. So that's tagging it for phagocytosis, so that means that it would act as an opsonin. Okay, why am I talking about complement now? Because antibodies work, one of the ways that they work is by, there's a binding site on an antibody for complement. And the fancy word they use is complement fixation. Or so they say that the antibody fixes complement. It means binds to it. Watch, I'm going to show you right here. Talks about it. Now look here. Through an antibody-antigen complex working with complement, it can cause phagocytosis, inflammation, and cell lysis. Anything that stimulates phagocytosis, once again, we can call an opsonin. You don't see that on here. So now you know, if you didn't spend enough time on complement and nonspecific immunity, you definitely need to spend some time on it. Okay? Generically, antigen, antibody complexes, which is the good guy and which is the bad guy? Antigen's the bad guy. We fight by using antibodies. It's the good guy. Look here. See this? A P? Right? It causes precipitation. What is precipitation? Yes, it is rain. We never see it, so we don't understand what that is. This is when you have, I don't know if you do this in AMP1 anymore. Anybody remember doing an experiment where you put little droplets of something in a clear liquid and then it turns cloudy? That's formation of a precipitate. It causes whatever that was insoluble, dissolved, to insoluble, not dissolved. Let me rephrase that. It's a soluble antigen. It settles out in the solution, is what we're trying to say. What I'm trying to say. Settles out in the solution. Well, if something settles out in a solution, that makes it a lot easier for phagocytosis to occur. That's pretty cool. Now, we said inflammation. We said antigen-antibody complexes, or antibodies, work by having a plan. There's the P in plan, precipitation. Agglutination. Look here. Which antibody is this? IgM. And notice if it binds a bunch of those, they kind of clump together. And it can create little cross bridges, too, between others. That's agglutination, or clumping together. That clump is easier to phagocytize. And, simply put, antibodies can cover the entire outside of a pathogen. And so they'll neutralize the pathogen, render it harmless, and of course, once again, that marks it for phagocytosis. So really, even though our book calls it the plan, I really think PAN works better because we don't see an L here, right? This is precipitation, agglutination, and neutralization. Down here is the PICL that we already have from complement. So they're just doubling up on the cell lysis part, really, is what they're doing. So we could say the complement part's OPICL, and the antibody part by itself is a PAN, but that sounds funny, so they put the little L in there for cell lysis, and they say that there's a little pickle plan or OPICL plan. Wow, that's crazy. Look, antibodies neutralize. They render substance harmless. Antibodies clump them together or agglutinate. Antibodies cause them to settle out or precipitate. Any of those makes it easier for phagocytosis to occur. That means that all of this together works as an opsonin, marking something or tagging it for phagocytosis. How many people are going to study up on complement now? Look here, neutralization, the simplest method. Antibodies block specific sites on a virus or bacterial exotoxin. Why? Because it's in body fluids. They prevent them from binding to receptors on tissue cells. They render them harmless. And then those complexes mark it for phagocytosis. What do we call anything that tags something for phagocytosis? Opsonin. Very good. Agglutination, cross-linking, binding, clumping. Precipitation, cross-linking that settles and is marked for phagocytosis, so we could call that also. It's an opsonin, or look, opsonization is taking place, tagging it for phagocytosis. Cool. All right, some more about antibodies. Wow. Antibodies are produced by what kind of cells? Okay, there's multiple answers here. Why? Because generically, B cells is absolutely positively the right answer. If I got very particular on a multiple choice question and you didn't see B cell, Sherry, what would you be looking for? Plasma cells are the particular ones that make it. But plasma cells are a form of a B cell. So it's okay if I just ask you what makes antibodies and you say B cell. That's right every time. However, if I ask you in a multiple choice question and don't put B cell there, you want to remember to look for plasma cells. Do T cells ever make antibodies? No way. They do not. They cannot. They make something. A monoclonal antibody, mono means one. One clone. That means that these antibodies, monoclonal antibodies, are prepared in a lab. They're commercially prepared. And they are only capable of recognizing one thing. Correct? That's why they're monoclonal. And this is brilliant. They take tumor cells, which are cells that grow too rapidly or divide too frequently or don't die off when they're supposed to, and they fuse them with a B cell. Well, a B cell is capable of making antibodies. They fuse it with a tumor cell, and then it makes even more. Wow. And it just dumps them out there. So they can produce these antibodies, single type of antibodies in a lab. And that is pretty fascinating that it can do that. And look, they're used in the treatment of certain conditions. They're also used in research. They're used in clinical tests. All right. This is how they do pregnancy tests to detect HCG. They prep monoclonal antibodies that would bind to HCG that's in your urine. You pee on the stick, it binds, and it makes a little positive most of the vaccinations, they put the bad guy in you. So they put the antigen or antigenic determinant in you. And usually it's dead, but sometimes it's live but neutralized, and they call it attenuated, and it's not supposed to be able to give you the disorder. They're learning even, that you can be too clean, right? All the antibacterial stuff's not necessarily good for us, just like they found out. And isn't this logical? Long ago, they used to give everybody antibiotics, right? And they found out, stuff becomes resistant. Well, guess what? We're causing stuff to become resistant on our skin from all the antibacterial products that we use. Oh my. It's a scary little bacterial world that we live in. Sometimes we're just in their world. There's a whole lot more of them than there are us. Okay, some STDs are diagnosed with monoclonal antibody tests, certain types of cancer, and like we mentioned, cancer treatment, especially like leukemias and lymphomas. Oh, and now it's time to talk about cell-mediated immunity. What cells? T cells. Let's look at a picture. Just to make sure you're really good and not confused. Hey, if you see a cell that looks like this, what's your brain think? Lymphocyte. By looking, you can't tell if it's a T cell or a B cell. That's the good news. I'm telling you, we're talking about T's now. Okay? We know that there's this little cell that we can call a CD4. Does anybody remember its other name? Very good. That's a helper T cell. And it's named that by its little receptor. Now we're going to say that this killer T, or here's the fancy name right at the bottom, a cytotoxic T cell, that's the main attacker of T cells. Remember how we called the B cell worker the plasma cell? The T cell component that's similar is a cytotoxic T cell. Well, didn't we say its job is to contact kill? And it's a cell and it's killing cells, so look, they call it cytotoxic. That means it kills cells. Cytotoxic Ts, because of their receptor, can be called CD8 cells. That's just memory stuff that I want you to learn. Okay? CD8. Can you see it up there? Right here. These are the cytotoxic Ts. They're also called CD8 cells. 8, as in the number 8, not gobble gobble. But that's a great memory device, even though they don't eat anything up. Because they're the killers. So look what we have. Where do the T cells mature? In the thymus. Look, they're showing us, look, this is the thymus. And then they mature in the thymus and they go out in the world. And they go to the lymphatic tissue where they get introduced to stuff. And that's where they meet up with usually their antigen-presenting cells, and they get activated. Well, we know that the helper Ts get activated and that they play a role in activating these others. We understand that, so this isn't showing us the whole picture. And then these cells spread out through the blood and go out through the tissues. Okay? And here's what we're going to do. And this is going to look very similar. What do we got? What do we classify it as? Antigen-presenting cell. An antigen-presenting cell has had contact with the bad guy. It's taken it in, and it's figured out what its surface markers are, and it displays them on the surface. Look, we could call that an AD. We would call this very good, MHC2. And we're going to say that this little guy, a T helper, comes in and contacts it. And what's the other name for the helper T? CD4 cell. And that helper T goes, here is the bad guy, right here. We can call a bad guy an AD. What would be a more generic name for it? Just antigen, right? And now we know what the bad guy is, and we want to stimulate some T cells. How are we going to do that? By releasing chemicals that we call cytokines. Now cytokines are out there. Now remember, this T cell, that is a nice healthy T cell, has had to have a prior contact. And this prior contact is way more important for T cells than it is B cells. Actually, some B cells can activate themselves without a prior contact and without going through that whole thing that we talked about. But I don't care that you really know that. I like to keep it as basic as possible. If this T cell has had a prior contact, it knows what it's going to be attacking, but it has to wait until the cytokines tell it to. So look, there's two parts, a prior contact and cytokines. So we say that in order to really truly activate these T cells, we must have had co-stimulation occur. Yes, they had their prior contact with what? The antigen. It's good enough with the antigen. And the cytokines from the helper Ts work together to activate it. So what can this T cell, this old generic T cell now do? It absolutely, positively is going to go through clonal selection. It is going to clone itself. Some of the clones are becoming memory cells. Will they be active now? No, they will lie dormant. Just very similar. Some of these cells, would it make sense that some of them could become T helpers? Sure. And we know what they're going to do. They're going to go out there and they're going to help. Aha. Many of these guys are going to become cytotoxic Ts, also called killer Ts. They will literally roam through the tissues, contacting our cells, looking to see if they're infected. I'll explain how that works in a second. We already hinted at it earlier in the night. Two others. Two others? No, one other. Then there's one group of cells that they used to call suppressors. I love that name, suppressor T. But now they call them regulatory, or Tregs, regulatory Ts, or Tregs. And think of it this way. T helpers activate, right? If we have a major activator, we also need to have something to shut the immune response off when it's time to shut it off. And that's what the regulatory Ts do. I liked the name suppressor because you didn't have to think regulates which way. But it's understood that T helpers are the activators, so the regulators are the shutdown cells. So, man. And there's some called delayed hypersensitivity cells, but we're going to talk about those later, okay, with a specific type of immunity. What, all of this, which cells are we talking about? T cells. And what type of immunity is the T cell immunity? Very good. It's called cell-mediated immunity. All we're going to focus on now is a killer T. And I don't even know if I need to draw a picture, honestly. We say that cytotoxic Ts kill with what's called a lethal hit. Remember, cytotoxic Ts are also called CD8s. A lethal hit involves basically two types of chemicals. The first chemical is called a perforin. You'll see it spelled in the picture as well. The second chemical is called a granzyme. Zyme means it's an enzyme. It can degrade the inside of the cell. Perforin sounds like perforate. It actually pokes holes in the cell. Cytotoxic. I won't abbreviate it like that, okay? But you can in your notes. It's a cytotoxic T cell. Grant, you'll see it again. Look right here. Here is target cell. No, that makes us think of hormones. Let's not think target cell like that right now. Here's a body cell, a normal human body cell. Except it's not normal. It's infected. Okay? It's infected. It's a normal body cell. It has an MHC what if it's a normal body cell? Number one. It's infected, so the protein is not self, the protein is foreign, correct? So now when the cytotoxic T comes over and contacts it, it says, MHC1, you're me. Bad protein, I need to kill you. How do I kill you? I inject my perforins into your membrane and create a hole or a pore or a channel. Through that pore or channel, I pump in little chemicals called granzymes. They break down the inside of the cell and cause the cell to die. What kind of cell is this that's going to die? It's one of my cells. It's my body cell. But it's one of my infected cells. Some bacteria can infect cells. Cytotoxic Ts could kill those. Usually, though, it's a virus. That's a pretty important mechanism for keeping infection in check when it's spread to our cells. Notice since the regulatory Ts slow down the immune response, they're important for slowing down or preventing autoimmune reactions. If we had the immune response not slow down, that would stimulate autoimmunity. Just like if too many of those T cells that got out attacked self. I think this is way too technical for just running through it all tonight. But this puts everything together. I would suggest that when you're studying, you just kind of look at it to get an overall feel, to remember what to keep separate, what's similar. Let's go back to this. I'm an infected body cell. Tell me what kind of surface marker I have. Surface marker first. MHC? 1. What kind of protein do I have displayed there? A foreign protein that's being displayed. So when a cytotoxic, I misspelled it, cytotoxic. When a cytotoxic T comes over, look what it's going to do. It's going to get really close there, and it's going to release its chemicals here and poke a hole. And it's going to pump other chemicals in here, and it's going to cause that cell to die. Remember, this is one of our cells that has been under attack by a virus, bacteria, or what have you. That's why they call these contact killers. They get up close and personal and kill that one. We see the picture right there. So now we just have to wrap it up. And here's how we're going to wrap it up. Oh, remember, we always talk about cytotoxic T's responding to infected self cells. It is also the kind of immunity that responds to transplants or grafts. Those are the cytotoxic T's. Cytotoxic T's. And tumor cells and cancer cells too. Tumor cells and cancer cells. Tumor cells and cancer cells are your body cells gone haywire. Cytotoxic T's take care of your cells that are infected, your cells that are growing too rapidly, cancer cells, or transplanted tissue that gets into us. And that is on a slide hanging out somewhere. Just don't know exactly where it is. So if you have a transplant, this is just little memory work. There's 4 types of transplants. If it's a transplant from your body to your body, like you want nice, puffy, cute lips, so they scrape some fat off of your backside and they inject it into your lips. You can do that. That comes from you. That's called an autograft. That is not an autograph. That is a graft from yourself. If you've lost some skin, they might take some skin off of you somewhere else and put it on you because there's no rejection from your own self, right? Isograft should not have rejection either because it's between identical twins who share the same MHC markers and share the same proteins because they have the same DNA that directs the building of proteins. So if you have an identical twin, a transplant should work out there. An allograft is between any 2 people. You would need to be on anti-rejection medication for the rest of your life. That's weird. Look, between any 2 people that are not identical twins. But that's kind of understood, just between 2 people that aren't identical twins. Because if you're a twin, an identical twin, it's called an isograft. And a xenograft is when we get a graft from a non-human. Pigs are some of the most common things for us to get grafts from. Heart valves. We can actually fit pig valves right into human hearts. And they work phenomenal. So they do it. You know what they're going to be able to do in the near future, though? You're going to be able to grow your own organs. They're going to be able to get our own stem cells. That's why cord blood is so important. If you have a baby nowadays, you want to save the cord blood because medical science is going to advance. And in that cord blood will be their stem cells. And they will be able to grow new livers and grow new hearts and grow new valves. And the thing about that kind of transplant, if it came from you, you would never need anti-rejection medication. So that would be pretty cool. Not quite there, but we're getting there. We can grow an ear on the back of a mouse. So, I mean, they can grow all kinds of stuff that we don't really concept that they're doing all the time. I mean, they can grow cells and grow a trachea now. They've grown a trachea and implanted a trachea that they fused with cells. They take and they strip out cells and they implant it with cells and it regrows. It's amazing what they can do now. Okay, just like always, there's some things you have to read. The immunodeficiencies, I want you to read about Hodgkin's and AIDS. Okay? The big bad thing about HIV and leading to AIDS is it attacks your helper T cells. It's the helper T cells that are wiped out by AIDS. And we understand, we get it now, that helper Ts activate your entire specific immunity. Yeah, it's cell-mediated immunity, but that can also mess with your antibody levels because part of the B cell response requires helper Ts too. Not all of it, but a huge part of it. Autoimmune disease, that's when your body attacks itself. Because it thinks it's fighting something foreign. Like lupus, like type 1 diabetes. Sorry, Nikki, I can't always say it the right way. Like, oh, I thought I had a nice little list here for you. Myasthenia gravis. Here it is, right in front of me. Multiple sclerosis. These are all autoimmune conditions or autoimmune diseases. That means that your T cells are destroying your own tissue. They're attacking your own tissue because in its head it thinks it's foreign or infected. For an arthritis, everybody should at least have heard of rheumatoid arthritis while you're in here and know that rheumatoid arthritis is an autoimmune condition. Your white blood cells, your T cells, actually destroy the joints, go in and eat out the joints. Commonly seen in the hands, and it causes the fingers to go like this. I think that in the near future, in big human history, in the next 50 years, medicine is going to be amazing. I think that what's happened in computers, that whole computer revolution in technology, is going to allow medicine to really skyrocket. And it's probably going to get away from just so much, taking a drug for certain things. They're going to go in new ways that we haven't even concepted yet. They're going to be able to, instead of taking a drug, they're going to be able to create these little chemicals that will put themselves together inside your body and go into the cells and operate in the cells and do things in the cells. I mean, it's going to be really fascinating what's going to be able to happen.